Well, good afternoon, everyone. I would like to start by thanking the Macular Society for funding this work and inviting me to speak to this exciting event. So, the research that we perform in our group is focusing at better understanding of eye diseases, this being inherited or age-related. We are located in this beautiful building named the International Centre for Life, next to the train station in Newcastle. To perform our work, we use several models, and this could be human eye samples that donated from the patients and their families upon consent, and the stem cells. So the human body contains many different types of cells, each with its own structure and features. A blood cell, for example, looks very different to a skin or nerve cell. But stem cells have the potential to change into more than one cell type, and that makes them extremely useful in research. So what are stem cells? These are rare and relatively unspecialized cells which have the ability to self-renew, that means produce another daughter cell, as well as develop into specialized cell type in the body. There are three types of stem cells, the first being embryonic stem cells. These are found in very early embryos and are pluripotent, which means they can change into any cell type in the body. Induced pluripotent stem cells are made in the lab by taking cells such as blood, skin, hair, and converting them to stem cells. Like the embryonic stem cells, they're pluripotent, so they can develop into any cell type. Adult stem cells, this supply the tissues and organs with new cells to replace those that are lost or damaged. These are multipotent, which means they can change into some cells in the body, but not any cell. In my talk today, I'm going to focus on induced pluripotent stem cells. With this approach, we can now take cells from adult patients and use to create cells and tissues that are usually very hard to observe, like the RP cells at the back of the eye, which are thought to be first affected by age-related macular degeneration. That means we can test now new treatment on cells that are genetically identical to those in the eye with someone with AMD, with no risk to the patient. Various pieces of research suggest that the retinal pigment epithelium, or RP as we refer to, is where macular degeneration begins. This pigmented layer of cells on the back of the eye serves as a pass-through between the light-sensitive photoreceptors of the retina and a layer of blood vessels called the choroid lying below. So our first funded project with Macular Society focused on generation of a lab model for AMD. In collaboration with ophthalmic consultant at Leeds and Northeast, we did take samples from several individuals who were diagnosed with AMD and had change in a key gene linked to AMD. We also took samples from several individuals without AMD. So one of the most significant findings in the AMD field has been the complement factor H, or CFH gene. And there is a small change, C to T substitution in this gene, which is strongly associated with susceptibility to AMD and has led to recognition of the importance of complement activation in AMD pathogenesis. So complement was discovered as a component of normal plasma that causes the killing of bacteria. And the complement system refers to more than 20 proteins which circulate in blood and tissue fluids. Most of these proteins are normally inactive, but in response to the recognition of molecular components of microorganisms, they become sequentially activated in an enzyme cascade. So the activation of one protein leads to activation of several other proteins in an enzyme cascade. Complement can be activated by 3 different pathways shown in this figure, and normally the key component activation here is the C3. So this can trigger the lytic pathway, which can damage the plasma membrane of bacterial and microorganisms. Complement factor H, the gene we're interested in this talk, serves as a brake, so it prevents the activation of the complement pathway downstream from C3. It is interesting that the complement factor H has strong binding capability to vertebrate cells, and this allows preferential protection of our cells as opposed to bacterial cells from complement-mediated damage. So what we did, we took blood and skin cells from the patients with AMD and the unaffected individuals and converted them to RPE cells. So we had three main questions in this project. The first one being, can induced pluripotent stem cell lines be established from AMD patients? Can those patient-specific lines differentiate to RPE cells? And then, can these patient-specific RPE cells be used to mimic disease and especially drusen formation? So in this following table, you will see the details of the individuals that did not have AMD, and we call them low risk. They have the normal sequence in the complement factor A gene. In red, you'll see the individuals that were diagnosed with AMD, in this case wet AMD, and they have the change in the DNA, so they have the C to T change in both copies of the genes. And we'll call these individuals high risk throughout this talk. So we were successful to derive human-induced pluripotent stem cells from all these individuals, and we differentiated those cells into RPE. You can distinguish these RPE cells here from the dark brown color. The RPE cells are beautiful. They're tightly linked to each other. They have this hexagonal morphology, and they have these very tight junctions to each other, shown here by this green staining. So once we derived the iPSCs from the low-risk control and the individuals with AMD, we wanted to make the RPE, which we did, and then to compare them. So what we saw when we compared the high-risk versus the low-risk cells is that the cells derived from the AMD patients were much more stressed. So you can see in this picture here, the cells contain those big vacuoles, which have the capability to burst the cell open. We also saw that the cell's mitochondria, the energy source, was affected. The AMD patients showed less mitochondria, and those who were larger in size. But importantly, there was a lipid buildup and drusen accumulation shown here beneath the cells with those yellow aggregates. So then we became really interested to find out how these deposits form, and so started to look at how the cells recycle their old and damaged components. The cells have a key process dedicated to this, and it's called autophagy, from the Greek word from self-eating. Basically, through the autophagy, the cells enclose the cytoplasmic components into those autophagosomes, and those autophagosomes then fuse with a different organelle called lysosome. So the lysosome acts more or less like a cellular stomach. It contains almost 40 enzymes that are able to break down either sugars or lipids or other waste products. So what we did see is that the cells, the RPE cells from the AMD patients, had the problem with those lysosomes, with those cellular stomachs. Those lysosomes were swollen, and moreover, the membranes were very fragile. They had pores from which the waste components could leak out and deposit into the drusen, so contributing even more to this drusen formation, making the cells unhealthy and not functioning well. So the rubbish was building up within the eye and causing the damage to the eye cells. So we were then very keen to find out if this buildup of waste was connected somehow to this uncontrolled complement activation. So we investigated several steps of complement activation, but here I'm just going to show only a quick photograph of the final step of complement activation, C5b9, which we refer to as membrane attack complex. This is a stable complex that mediates the irreversible cell death. We found that more of this complex was internalized in the RPE cells of high-risk donors. So the high-risk donors shown here on the right and the low-risk donors shown here on the left. But what was really interesting is that this deposition was actually happening at the lysosomes. So again, we could find a marker that did detect the deposition at the lysosomes, the low-risk donors being in the left and the high-risk donors being in the right. And you could see easily from this picture that there is more deposition at the lysosomes in the RP cells from the AMD patients. From these findings, we then hypothesized that uncontrolled C3 turnover is the upstream cause leading to dysregulation of the waste disposal in these patient cells. So to prove this, we needed an inhibitor of the C3 activation. So in collaboration with Professor John Lambris, we got hold of a compound named CP40, which has got strong and exclusive affinity for C3. And this prevents the critical step that is essential for initiation, amplification, and the terminal pathways of complement. We found that application of CP40 for 7 days reduced this uncontrolled complement activation, maintained the lysosome structure so they were not leaky any longer, also maintained RPE function, but moreover reduced the drusen deposit. So here now on the left, you again see patient cells. They have lots of this final membrane attack component in the lysosomes. However, when we treat them with a C5b9, you could see that this deposition is much reduced. Importantly, the drusen deposit, again shown here as yellow aggregates, are present in the patient cells, but they reduced in size upon application of this component CP40. So we were actually very happy that the application of CP40 seemed to bring cells to a functional ability as in the controlled individuals. To date, CP40 has shown promising therapeutic efficacy in non-human primate models of kidney transplants and hemorrhagic shock. And recently, a successful single phase 1 trial with a CP40-based drug candidate has been recently completed in healthy male volunteers, showing very good safety profiles. So collectively, this finding suggests that an inhibition of C3 turnover with CP40-based drug candidates may provide an effective therapy for patients with AMD. However, further studies looking at the route and frequency of delivery, as well as safety and efficacy, are needed to implement these research findings into treatments for AMD. So once we established that RP cells from AMD patients were not able to get rid of the cell waste efficiently, we asked the question if they developed other ways of getting rid of waste material. And for this, we looked at the nanoworlds. So cells contain organelles that are released in the cellular microenvironment. And these are shown here in yellow. These tiny microvesicles travel between the cells all the time to help repair damaged tissue, grow new blood vessels, perform wound healing, and encouraging blood to clot around an injury. Incidentally, viruses can also use exosomes to spread from one cell to another. So our second project with the Macular Society focused on 3 key questions. Do the AMD RP cells produce more of these vesicles? Secondly, is the content of these vesicles different to those purified from individuals without AMD? And thirdly, are these vesicles important for the pathogenesis of the disease? So the answer to the first question is yes. We did purify these tiny bubbles or microvesicles from RP cells of patients with AMD and individuals without AMD. And as you can see in this figure, the RP cells taken from patients with AMD produce almost double of those tiny vesicles. So now knowing that the RP cells of high-risk individuals produce double of these vesicles, we just wanted to compare them. So these vesicles carry a cargo inside. So we wanted to compare the cargo of the tiny bubbles from patients with AMD and individuals without. And by doing that, we found that the cargo contained genes that were related to cell stress, changes in cell architecture, and also genes that encourage new blood vessel growth, which corresponded quite well with the clinical diagnosis of those patients having wet AMD. On the bottom side, those cells were producing also tiny vesicles, and their cargo also carried genes that were important in blood vessel remodeling and changes in cell metabolism. So the question is, can now these changes be made into something useful? Can they be used for diagnosis? So we now have a third project funded by the Macular Society. And this is a collaborative project with Professor Andy Lottery and his group at the University of Southampton. Over the years, they have successfully managed to control the blood plasma samples from AMD patients, and these are about 409 samples, and unaffected individuals, so this comes from 322 controls. Using our cell knowledge and the work we've done so far, we found about 8 different candidate proteins that are more highly expressed in those tiny bubbles coming from the AMD patient compared to unaffected controls. So our plan for this project is to use first those 8 candidate proteins in a limited number of samples. So first 23 samples from AMD patient and 22 from controlled individuals. Our preliminary data seems to suggest that this is a very powerful approach. For example, you'll see here one of the candidate proteins is not expressed in the plasma samples of unaffected individuals, is highly expressed in patients with dry AMD, but not present in patients with wet AMD. So once we've done this work, we then want to take 2 of these top markers and validate them in the larger patient samples that we have. The idea behind this is to come with 2 or 3 diagnostic markers that can be used in plasma, in blood samples, and then maybe hopefully diagnose the disease before we reach to the end phenotype, which is loss of vision. We're also interested to know if these microvesicles had a function. So obviously the cells are talking to each other. They're using those microvesicles as cell, but does this talk have a meaning? So to do this experiment, we again group RPE cells from patients with AMD. And then we purify those microvesicles. So those purified microvesicles were then added to RP cells for control patients for 14 days. So our idea is that if they had a role, that would make the control RP cells sick. And this is exactly what happened. So the control RP cells, without those tiny vesicles, were nice. They had those melanosomes, which produce the pigment. However, when we added the tiny bubbles from the patient AMD RPE cells, we again saw signs of cell stress, exactly as the patient cells themselves, and the increased numbers of those lysosomes, which are now again becoming fragile and were unable to function properly. But we know that the RPE cells are also in close contact with photoreceptors. So we were able in the lab to make mini retinas or retinal organoids, and they have beautiful aligned photoreceptors on the outside. So instead of having the RPE cells here, we added those tiny bubbles produced by the patient RPE cells, again for 14 days. And what we did see, there was a specific cell type called the Müller glia cells, which are very important for the function and the stability of the retina. They became swollen and looked stressed, and in some cases they also look very much like retinal photoreceptor progenitors. And this is not surprising. We know that in vertebrate species, but not humans, in response to acute injury and stress, the Müller glia cells do transdifferentiate to retinal progenitor cells. So we do believe that actually the tiny bubbles or vesicles provided here by the patient RPE cells are causing stress also to photoreceptors. And the first acute stress that we're picking is in Müller glia cells. But this is an ongoing project. And we would like to investigate not just acute stress, but would like to investigate how chronic stress or continuous stress then leads to photoreceptor damage. So to just summarize what we found in this ongoing project is that the patient cells have a defect in the waste disposal, and is mainly located at the lysosomes. So they're fragile, they're not functioning properly, waste is not being disposed, but instead is accumulating in those drusen deposits. So to compensate for this, those patient RPE cells are now producing those tiny bubbles of vesicles that carry a cargo, But this cargo is also diseased in a way. It's carrying genes and proteins that act as a disease messenger when they reach the next cell that is healthy. So that means that those tiny bubbles are creating a vicious circles that propagating the disease in other cells. But moreover, they're not just making the RPE cells ill, they're just also making the photoreceptors disease, which explain why eventually loss of vision happens. So if we can improve this RPE cell waste disposal process, we could prevent not only the buildup of damaged waste material, but also the spread of inflammation to other unaffected cells. And spotting exosomes with the hallmarks of AMD could help us diagnose it earlier before a patient notices any change to their vision. So I would like to finish here by again thanking the Macular Society for funding our work, the patients for donating their tissues, and all the researchers and clinicians involved in this project. In particular, Ed, Marcina, and Dean have spent the last 7 years working hard on this project, and it couldn't have been here without the help of our clinical colleagues, such as Professor Still, Professor Lowtery, autophagy expert Dr. Korolchuk, IPS expert Professor Armstrong, and a complement expert Professor Harris. So thank you for your attention, and I'm happy to take any questions.